Okay, so we're going to carry on with where we left off yesterday. So I sort of covered a bit of diffusion, a bit of osmosis, a bit of facilitated diffusion, and a bit of active transport. So we sort of left it on this exam question, which was an example of one that comes up quite a bit in the exams. So I'm just going to have a talk through the answers for this. So some molecules move across a cell surface membrane by passing down a concentration gradient through the phospholipid bilayer. The movement of some polar molecules across the membrane involves carrier and channel, and these would be protein molecules or glycoprotein molecules. When this movement occurs down a concentration gradient, the process is called facilitated diffusion. And when it occurs against a concentration gradient, this process is called active transport. Energy in the form of adenosine triphosphate, ATP, is used in the movement of molecules against a concentration gradient. Now, the biggest issue that people come up with when they answer this question is this one here, where it's facilitated diffusion. So when movement occurs down a concentration gradient, the process is called, and what we often find is lots of people put diffusion here, or sometimes they actually will even get to the point of writing osmosis. But we've actually said up here, we've mentioned the carrier and channel proteins, the glycoproteins. So we've mentioned those carrier proteins. So obviously normal diffusion would happen through the phospholipid bilayer. The polar molecules are going through the channel proteins. So therefore we're talking about facilitated diffusion, or if we're going against concentration gradient, active transport. So that's something that's really important to remember, is actually that very fine difference between facilitated diffusion and normal diffusion, simple diffusion. So exocytosis and endocytosis. These are two new sort of terms. They're actually quite self-explanatory, but quite nice actually. So exocytosis is the removal of molecules from inside the cell. And this is actually when we're going to have large molecules, large particles, large amounts of them actually being moved out of the cell. It's not very, what is the word I'm looking for? It's not very efficient to be using active transport or protein channels for things like proteins, polysaccharides, insulin, vesicles. So actually all these things actually want to leave by exocytosis. And exocytosis basically is we have a secretory vesicle, which is made in the Golgi apparatus, with whatever is inside the middle of this secretory vesicle that we're going to excrete. So for example, insulin. So this could be insulin inside the here. Basically the secretory vesicle is just made up of another type of phospholipid bilayer, but it makes that sort of circular form. It comes and it fuses with phospholipid bilayer, so it actually breaks the bilayer apart because it's made just of more phospholipids, and then it releases these materials, so for example the insulin, out of the cell. So for example, it's really good when we're wanting to move huge quantities through. So that is what happens with exocytosis. Endocytosis is basically just the exact opposite. Endocytosis is when things are being taken into the cell. So things like large molecules from outside the blood membranes, so things such as white blood cells when they engulf bacteria. So that whole process of phagocytosis, that is a type of endocytosis. Exocytosis, a good example of exocytosis is neurotransmitters being released from neurons across the synaptic cleft. So that is a good example of exocytosis. And then endocytosis, a really good example is white blood cells engulfing pathogens in phagocytosis. So another quick overview. Again, we've got our transported molecules. They can either get into our cells via passive transport, either through simple diffusion, so straight through diffusing through the phospholipid bilayer, or using facilitated diffusion and they go through the carrier proteins. Or if they're going against our concentration gradient, we're going to use active transport and we'll go through a protein pump, a protein channel, and energy is required, and in this form it is ATP. So I'm just going to have a little talk about diffusion and what affects the rates of diffusion. So we mentioned Fick's law before, and Fick's law is basically looking at the concentration gradient, the thickness and the surface area. So simple diffusion, and simple diffusion is basically just diffusing through a membrane, so we don't need any proteins, any channel proteins or anything. So simple diffusion, it depends on three things. So the first one is concentration gradient. The higher the concentration gradient, the faster the rate of diffusion. So that's sort of the main thing to remember. So basically what happens is, as diffusion is taking place, the difference in concentration between the two sides of the membrane decreases until it reaches equilibrium. So obviously it's not going to maintain that really, really high difference. It will go down, go down, go down, and go down as more of those molecules are diffusing into the cell until we reach equilibrium, i.e. the concentration on both sides of the membrane at that point is then equal. This means that diffusion slows down over time. It would be, unless we are constantly supplying the molecules or whatever the substances that's going to diffuse through the membrane, unless we were supplying it ourselves constantly, so giving it a constant supply, and it is basically a limiting factor, it will eventually slow down. So that concentration gradient will decrease, and eventually the rate of reaction, the rate of diffusion will slow down over time due to concentration gradient decreasing over time. Another one is the thickness of the exchange surface. So the thinner the exchange surface, so i.e. the shorter the distance the particles have to travel, the faster the rate of diffusion. Smaller distance, they're going to travel faster. And then the last one is surface area. So the larger the surface area, for example, the cell surface membrane, the faster the rate of diffusion. So we want a large surface area, we want a high concentration gradient, and we actually want a very small thickness, very small distance to travel to get rate of diffusion as high as possible. This is for simple diffusion. Now on here I've got an image of the lumen of small intestines, and small intestines are made up of villi, and actually then when we look at our villi, we actually have microvilli on them. Microvilli increase the surface area of the small intestine for faster diffusion. So some cells, so for example epithelial cells in the intestine, there's other cells that have them as well, have these microvilli. And these are projections formed by the cell surface membrane folding up on itself. So it's just the membrane folding up on itself. So it could be really, really long, but it would take up lots of space. So it actually just folds up on itself. And the microvilli give the cell a larger surface area. So it's got a large surface area for the space that it takes up. In humans, microvilli can actually increase the surface area by about 600 times. So if it was just a straight flat line, the surface area would be 600 times smaller. So by having these projections, it increases the surface area 600 times. And a larger surface area means more particles can be exchanged in the same amount of time. Therefore, it increases the rate of diffusion. So it's a bit, so it's not, so it doesn't make it, it makes it faster because we increase the rate of diffusion, we're increasing how many particles can diffuse. The things that affect, factors that affect the rate of diffusion for facilitated diffusion is actually slightly different. So we're just going to look at them now. First one's concentration gradient, stands the same. So the higher the concentration gradient, exactly the same as before, the faster the rate of facilitated diffusion, up to a point. And I'll explain that up to a point again. As equilibrium is reached, the rate of facilitated diffusion will level off. So very, very similar to simple diffusion. Now, this is the main difference here. So here because we're using channel proteins, thickness of the membrane, of the exchange surface and surface area isn't affected here. What actually is now, it's a number of channel or carrier proteins is what affects it. So the number of channel or carrier proteins, so once all the proteins in a membrane are in use, facilitated diffusion cannot happen any quicker. So for example, in this image, there's 4 channel proteins there. Once all four are being used, diffusion, the rate of diffusion cannot increase. So even if we increase the concentration gradient and we just, for example, we've got water here, we just stuck more water in there and more water in there and made that concentration gradient higher, it cannot increase anymore if all of the channel proteins are in use. So the greater the number of channel or carrier proteins in the cell membrane, the faster the rate of facilitated diffusion. So actually for facilitated diffusion, our concentration gradient, whilst that is true, that it has a bit of a caveat along with the channel and carrier proteins. So having more channel proteins increases the rate of facilitated diffusion. So we've got an image here of an aquaporin, and aquaporins are special channel proteins, and these allow facilitated diffusion of water through cell membranes. Some kidney cells are actually adapted to have lots of aquaporins, and aquaporins, what they do is they allow the cells to reabsorb a lot of water that would otherwise be excreted by the body. And what they do is they actually reabsorb 180 litres a day. Water can diffuse directly through the membrane, even though it's polar, because it's relatively small, but actually the aquaporins help us be able to take in even more. What you can get asked in the exams is sometimes you might be asked to calculate the rate of diffusion from a graph. So if it was a straight line graph, for example, you would need to find the gradient of the line. For a curved graph, it would mean finding a tangent. We'll look at those sort of skills in another time when we look into graphs a little bit more in detail. Osmosis, the rate of osmosis and how to do osmosis is actually a practical that you need to know. So I'm just going to get this other PowerPoint up for you. I'm going to give you an example of one of these practicals. So osmosis. We know osmosis is this fusion of water molecules across a partially permeable membrane from an area of high water potential to an area of lower water potential. Now, the rate of osmosis, if this would actually open properly, the rate of osmosis depends on three things, and you'll actually realise that these are very similar to diffusion. Water potential gradient, basically concentration gradient, but obviously with osmosis we're only talking about water, so therefore instead of saying concentration gradient we say water potential gradient. The higher the potential gradient, the faster the rate of osmosis. Osmosis takes place, the difference in water potential on either side of the membrane decrease, so therefore the rate of osmosis levels off over time the same way that diffusion does. The thickness of our exchange surface, exactly the same as simple diffusion. The thinner the exchange surface, the faster the rate of osmosis. And then finally, our third thing is surface area. The larger surface area, the faster the rate of osmosis. Now, you can get asked to talk about different types of practicals, finding water potential and membrane permeability, and different things. So this is one example to assess if alcohol concentration has an effect on membrane permeability. So obviously we talked about membranes being permeable, we talked about cholesterol, and then a practical might be to see how alcohol actually affects this membrane permeability. So I'm going to talk through actually what you could do to find this out. It's exactly the same process as if we were changing it with temperature. So alcohol or temperature on membrane permeability. And there's a couple of different techniques that we need to be able to talk about and use as well. So a massive thing, and you'll have seen this at GCSE as well, is how to make our results reliable and accurate. You can get asked to write methods in the exam, and you can get asked to show that it would be precise, reliable, accurate. So precision is the erroneous deviation from a constant as measured under conditions where no deviation is a possibility. basically, precision is when all of our results are similar. Doesn't mean necessarily that they're to the true value, but they are all precise. They are all very precise to each other. So reliability. Reliability is the extent to which the measurements of a test remain consistent over repeated tests of the same subject under identical conditions. So an experiment is reliable if it yields consistent results of the same measure. So reliability is a very strange one. It gets mixed up with reproducibility a lot, but basically reliability is if you've set out a method and you did exactly the same thing again the next day using the same equipment, using the same measurements, doing everything exactly the same, you should be getting consistent results. We obviously know there could be some errors here and there, but you should be getting consistent results that are similar. So that is the reliability. So you can get asked things such as, how can you ensure your experiment is reliable? This is where you would need to discuss actually making very precise methodology. Actually, this would be where you would bring in, well, actually repeating your experiment, not just using one set of data, just doing everything once. You actually need to show that your experiment is reliable, your results are reliable, by actually carrying out your experiment realistically at least probably 6 to 10 times. You can say 3 times because you can take an average with the 3, but it's not the best. It's the more replicates that you take, the easier it is to find an anomalous result. So that's where we sort of are with that one. Basically, what you would do is, you would get different amounts of, you'd cut up some beetroot or potato. Beetroot's a bit easier, and I'll explain why we would use beetroot. And you would get different amounts of dilutions of alcohol. So for example, the different percentages of ethanol. One thing that you have to be able to do, and I'm going to swap to a different screen here, is you would need to be able to talk about serial dilutions. So investigating water potential or membrane and permeability using something called a serial dilution. And this is what I'm going to talk through quickly with you now, is what a serial dilution is. Serial dilutions are basically when you start off with one concentration of a liquid and then we make them stronger or weaker as we go. But with it being dilution, we make them weaker as we go. So let's just move this out the way a little bit. And let's pop this here. So we've got serial dilutions. Serial dilutions are something that you can get asked about a lot. You can be expected to explain these in an exam. I'm just going to talk through the processes of what a serial dilution is. So let's go back to a very typical water potential experiment, osmosis experiment using potatoes. You've probably done this one at GCSE where, let me just get another page up for a second. You would have a potato, you'd cut bits of it up, and then you'd have a load of test tubes, and you'd put bits of potato in these test tubes, and they'd be put in test tubes that've got different solutions in them. So they could be different solutions of water with a different sugar concentration or salt concentration. So we could say this was 10%. We'd probably have a 0 as well, so let's stick a 0 there. 25%, this is a bad example, 35%. So we'd have all different concentrations of salt solutions. You'd leave the potato in there, and then we'd see what would happen to that potato. We'd check the mass of that potato afterwards, and it's all about finding out where the water potential is. So that's where you would then do the graph, and we would then plot the water potential on. And we'll look into that in a bit more detail in a minute, but that's sort of to reignite your memory of that experiment. Now, GCSE, you are fine to just be given all your different dilutions. You're fine to be given a solution of 10%, 25%, 75%, 0%. At A level, you're expected to be able to make up these serial dilutions yourself and actually explain these serial dilutions yourself. So that's what we're going to talk about now. So you need to make up several solutions of different known concentrations. So that's really important. And you can use this using the serial dilution technique. Now, you don't have to dilute solutions by a factor of 2. What we normally do is like factors of 10. So I'm going to just show this one to begin with. So often one we would use is a serial dilution, 5 serial dilutions. So of the sucrose solution. So obviously sucrose is sugar. And we're going to start with an initial sucrose concentration of 2 moles. And we're going to dilute it each time. So we're going to do 5 serial dilutions. So 1, 2, 3, 4, 5. Oops. Hi. Okay, so we're starting off with 2 moles in this one. So this is a sucrose solution. So it's a 2 molar sucrose solution. I'm just going to write this over here, sucrose solution. And basically we start off with our original 2 moles and we dilute this down each time. So what you would do is you would line up 5 test tubes in a rack. This is 5 test tubes. I'm going to badly draw a test tube rack to show that they're all lined up in a rack. And there we go. That's test tubes in a test tube rack. Let's maybe make, I can't bother to make that test tube longer. Okay, so then what we would do, put our test tubes in the test tube rack. We would add 10 cubic centimeters. So I'm just going to put an arrow in here and say how much is in there. So we've got 10 cubic centimeters of the initial 2 mole sucrose solution to that first test tube. So that is of our 10 cubic centimeters of our sucrose solution. And that would go straight in there. So that's 10 centimeter cubed sucrose solution straight in. Then we are going to then add 5 centimeters of distilled water to the other 4 test tubes. So this would be stage 1. Stage 2, I'm going to just sort of colour these in slightly differently. We're going to add 5 cubic centimeters of distilled water to these ones. Okay, so imagine we've got 5 cubic centimeters in. I'm not going to draw the solutions in at the minute for a reason. Okay, so now using a pipette, you will draw 5 cubic centimeters of the initial from the first test tube and add it to the distilled water. So what we would do is we would take 5 cubic centimeters. Yeah, I am right. 5 cubic centimeters of that initial sucrose solution, and we would put it into this test tube, into the second test tube, and we would mix it thoroughly. So then in here, we would have a solution that's got 5 centimeters of 2 mole solution, 5 centimeters distilled water. So this would actually come in at a total then now, and it would become a 1 mole solution. Okay, so this would then be a 1 mole solution. Now it's a serial dilution. You can probably see where we're going with this process. Mix it thoroughly. We now have 10 centimeter cubic solution, cubic centimeter of solution that's half as concentrated. So then we repeat this process. So actually what you should end up with is we then take 5 cubic centimeters out of there, put them into the next one, and then our next one would end up as a 0.5 mole solution. We take 5 cubic centimeters, oops, I don't know why I wrote 0 there. We take 5 cubic centimeters out of that one, mix it thoroughly with the water, and we'd end up with a 0.25 mole solution. And then finally, we take another 5 cubic centimeters out, and we would get a 0.125 mole solution. And so that's a zero dilution. You don't have to dilute to factors of 2. You can dilute to 10s. So for example, if we were doing it to a solution of 10, let's put some test tubes here. If we had an initial 10, let's put that 2 mole. We had our starting of 10 cubic centimeters in here, and rather than taking out 5, you could take out 1 cubic centimeter, and then you could take out 1 again, and so forth. So you can change whatever rate that you're diluting by, but that's how we can do that. So that's one way of making different solutions of different concentrations, zero dilution. So we start off with a really high concentration and then we dilute it down. There is another process for doing this called scale factors. So this is when we want to make a sucrose solution. Well, it could actually be a salt solution. It doesn't actually have to be sucrose, but we're going to use sucrose again, of any concentration, and we're going to use a scale factor. So the issue with zero dilutions is it obviously has to, we're just diluting it each time. So we're just going down each time by a set amount. So for example, we're going down by factor of 2 the first time, so it needs to go down in factors. This is easier. Scale factors are if we want to make sucrose solutions for any concentration. So for example, if we wanted to make a sucrose solution that was 15 cubic centimeters, but we want it of a 0.4 mole sucrose solution. So here we've got a different volume than initially, and we've got a different concentration. So start off, we're going to start off with a solution of a known concentration. So we have a starting solution of a known concentration, starting solution with a known concentration. So for example, it could be a 1 mole. So let's start off with 1 mole. The second thing you do then is we're going to find the scale factor. Okay. And what we do is we find the scale factor, and we do this by dividing the concentration of the solution. So dividing the conc of the solution by the concentration of the solution we want to make, by the concentration of the solution we want to make. So for example, in this situation, we've got 1 mole. We want to make 0.4 moles, so we do 1 mole divided by 0.4 moles, and that would give us 2.5. So this 2.5, this means the solution we want to make is going to be 2.5 times weaker than the original, than the 1 mole, than the 1 mole. So to make the solution 2.5 times weaker, we use 2.5 times less of it. So for example, if we've got 15 cubic centimeters, we would divide this. So we've got 15 cubic centimeters of 1 mole. We divide 15 cubic centimeters by 2.5 because we want it to be 2.5 times weaker. And that would mean we have 6 cubic centimeters. So we would then transfer 6 cubic centimeters of the 1 mole solution to a clean test tube. What we would then do is make up the concentration with then distilled water. So our total concentration we want is 15 cubic centimeters. So we want a total volume of 15. We've got 6, so we do 15 minus 6. That gives us 9 cubic centimeters of distilled water that we need to add. So this is a scale factor, and this is how we can make up a solution of any concentration at any volume using a scale factor. So you could do this for anything. We could say pick any values, and this is how you'd work it out. So this is how we can do this by using scale factors. Zero dilutions are a little bit easier because we're just doing the same thing again and again and again, and it's getting weaker. But obviously, if we want quite large quantities and we want them to be of quite varying different concentrations, and maybe it's quite hard to do that by using serial dilution, then we can use scale factors instead. So I'm going to go back to sort of where we were talking about that potato experiment, like what you did at GCSE. We have to know it again at A level. It's actually exactly the same method, but we're going to talk through it again. So back in GCSE, you cut up potato to see what the water potential of it was. Same thing's going to happen again, A level. Same method as well. So the first thing you're going to do is you're going to cut up your potatoes. You're going to use one of those cork borers you use at GCSE. They're those sort of like metal cylinders that just go and bore into potato. Now you think, why do we use one of these rather than, for example, just using a ruler and just cutting up a bit of potato? Well, we use a cork borer for that precision. The idea is that is a lot more precise than us just trying to chop up a potato with a knife. So it gives us that precision, and it gives us identically shaped pieces of potato. If we're just trying to use a knife, they're not going to be identically shaped. They're just going to be all over the place. So we use a cork borer because that is going to give us identical shaped bits of potato. And then we use a ruler to chop them into 1 centimetre diameters. So then roughly, we should have roughly about the same amount of potato there every time. Obviously, we don't know if that's going to work. So what we have to do is we have to record the mass. So we actually divide the bits of potato up, and we are actually going to do them in groups. So we divide the potato chips into 3 groups. So cork borer, and we're going to call chunks of potato chips now. So we're going to put the chips into 3 groups. And then we're going to measure the mass of each group. So the mass of each group using a mass balance. So set of scales, get the mass of each group of potato chips. What we would then do is we would place one group into each of our sucrose solutions. This is where it becomes debatable. Some people say, yeah, it's fine, we can leave them for 20 minutes. Some people say 24 hours. I'd say definitely more than 20 minutes. Most of the time, to be realistic, 24 hours is probably the best to leave. But the most important thing is that it's all left for the same amount of time. You're going to then dry them with a bit of paper towel. And these are the different sucrose solutions that you've made using your serial dilution or using your scale factor. And then number 6, we're going to record the mass, and we're going to find out the change in mass. For each group. And then what you would then do is you would use your results to make a calibration curve. Oh, calibration curve showing the change in mass against sucrose solution. So I'm going to show you what a graph like that would look like. So let me just draw you this on here. So along the side, we would have percentage change in mass. Let's do this as 10, 20, minus 10, minus 20. And then across here will obviously be our concentration of sucrose solution in moles. Sucrose solution, and that will be in moles. So we might have one at 0.2, oops, 0.25, 0.5, 0.75, 1, 1.25, 1.5, 1.75, and then 2. Okay, now we did 5 serial dilutions. So we're going to use those same 5 serial dilutions that we actually had initially. And if we remember back then, our solutions were actually 2 mole, 1 mole, 0.5 mole, 0.25 mole, and then 0.125 mole. So let's put the results on. So let's say, for example, our 0.125 mole, the change in mass was about 19. Percentage change was about 19% positive. Our 0.25 mole, the change in mass was about 2% positive. 0.5 mole, the change was actually about minus 8%, and then our 1 mole was about minus 14, minus 50, so let's pop that there, and then our 2 mole was about minus 80, or no, it was basically minus 20, so let's put it on minus 20. Then what you would do is you would draw the curve. Now it can be quite hard for me to try and do this on the computer, but let me have a go. Yeah, no, that did not work. So let me just try one more time. Try and get this to work. And you want to try and get it through points as best you can. Now, I am just going to, what I would say is don't do what I'm about to do, which is rub it out after you've done it, because that was not what I was intending to do. Let me get a different colour. Now, obviously, a curve is supposed to be one continuous line. It's not a dot to dot. But yeah, there we go. I've gone through the points as best I can on a computer. Now, a really important thing on here is this point here. And this is the word isotonic that you've probably heard of before. So this point here, so we'd probably say that's about 0.27, 0.26. Let's call it 0.27. 0.27. Okay, we'd say this point is where our water potential of the sucrose solution is the same as the water potential inside the potatoes. So water potential of sucrose, same as potatoes. So this is our isotonic point. You can regularly get asked to show an isotonic point. So you can be given a set of data, asked to draw your own graph, draw the graph, draw your calibration curve, and then find the isotonic point. So potato chips will gain water and therefore mass if the solutions with a higher water potential, in solutions with a higher water potential than the chips, and they will lose water in solutions with a lower water potential than the chips. The point at which the curve crosses the x-axis that we've just talked about, where the percentage change in mass is 0, is a point at which the water potential of the sucrose solution is exactly the same as the water potential of the potato cells. We find the concentration at this point, and then we look up the water potential for that concentration of sucrose solution. So we would use a textbook to do that. So you wouldn't be expected to do that in an exam, or they might give you that on an exam. What we're going to just have a little last talk about is, I'm just going to get an image of a mammalian ileum for you. So let me just, oops, I don't know what I've done there. Give me 2 seconds. Mammalian ileum. Actually, no, I'm just going to have that drawing this instead. Where's the whiteboard? Ah, here we go. So the last little bit we're going to do is just have a little talk about active transport. So active transport, we covered most of that yesterday, and we're going against concentration gradient. It requires energy. It's going to use carrier channel proteins. Things that affect the rate of active transport. We've actually got 3 things that affect the rate of active transport. So I'm going to put AT here for active transport. There's 3 things that affect the rate of active transport. The first one is our speed of individual carrier proteins. Now, carrier proteins have different speeds. So when we think back to when we're talking about facilitated diffusion, if all those carrier proteins are being used, rate of diffusion can't increase. The rate of active transport relies on the speed of those individual carrier proteins. So the faster they are, so the faster they work, equals the faster the rate of reaction, equals the faster rate of active transport. Second thing is our number of carrier proteins or channel proteins. Exactly the same issue as when we were talking carrier channel proteins, when we were talking about facilitated diffusion. Once all of them are used up, active transport cannot increase. So the more of them we have, the faster active transport can take place. And then finally, this one's very different from anything in diffusion. The rate of active transport relies on the rate of respiration. Now, that might seem a bit obscure at first, and thinking, well, why does respiration involve? Well, that's obviously because the rate of respiration and the availability of ATP. Now, obviously, active transport is not a passive process. It requires energy. Therefore, if respiration is inhibited, active transport cannot take place. So we need a high rate of respiration, therefore that ATP can be produced, so that ATP can then be used for active transport. And the last thing we're going to look at is the mammalian ileum. So glucose is absorbed actually by co-transport in the mammalian ileum. So I'm just going to talk about that now. So glucose is actually absorbed into our bloodstream in the small intestine. We're sort of getting to that part of our body. So the ileum, and the ileum officially is the final part of the small intestine. The concentration of glucose is too low for glucose to diffuse out into the blood. And this is that important part, actually. Basically, glucose enters our bloodstream using diffusion, apart from when that concentration gradient has eventually petered out and we get to that point actually where diffusion can't take place, where it's got to that equilibrium point. And that happens once towards the end of the small intestine at the ileum. So Glucose is absorbed from the lumen of the ileum by co-transport. So it uses active transport. So this is what we're going to have a little look at now really quickly. So I'm just going to draw you a little image of the lumen and the ileum. Oops. Oh no. Right, just give me a second, sorry. Okay, let's get rid of that last one actually. I don't need that. So I'm going to draw on some proteins. And this is going to be a channel protein. I'm going to colour that in. So up here we have got blood down this side, and we've got our lumen of our ileum up here. Lumen of ileum up here. Okay, so glucose enters the ileum epithelium with sodium ions. So this is the thing that we mean about co-transport. It doesn't just get in there by itself. It actually uses something called co-transport. So down at the bottom of our, down here, we're going to have some other proteins. Okay, so sodium ions, what happens is sodium ions, so we've got potassium, let's get potassium coming in, and we've got sodium. So number one, sodium ions are actively transported out of the ileum into the blood. So let's show that. We've got sodium ions being transported out, actively transported out into the blood by the sodium-potassium pump. And this is what I was saying earlier. Some pumps can just be for one type of ion. Some pumps can use multiple ones. So first of all, sodium ions are actively transported out of our ileum epithelial cells into the blood by the sodium-potassium pump. What this does is this establishes a concentration gradient. There's now a higher concentration of sodium ions in the lumen of the ileum than inside of the cell. So we've got basically more sodium up here than in here, which is good, okay? So what this does is this causes sodium ions to diffuse from the lumen of the ileum, so this is number 2, into the epithelial cell. So we've got coming in, let's have this coming in here. And I've just used the word, so actually I'm just going to pop over here that this one is, so number 1 is using active transport. So 1 is AT. Number 2, so causes sodium ions to diffuse. So this is obviously going to be facilitated diffusion because we're using a channel protein. So it causes sodium ions to diffuse from the lumen of the ileum into the epithelial cell, down their concentration gradient. They do this via the sodium glucose co-transporter proteins. So this is the sodium glucose co-transporter protein. Number 3, the co-transporter then carries glucose into the cell with the sodium. So let's pop that there. I'm just going to write glucose underneath it just because I haven't got as much space above. As a result, the concentration of glucose inside the cell increases. So 3, co-transported glucose with sodium. Therefore, glucose will then diffuse out of the cell into the blood, down its concentration gradient, through a protein channel by facilitated diffusion. And so then this is protein channel. That's going to be our glucose. And that's facilitated diffusion, So overall, we would call this active transport, but actually it's active transport with facilitated diffusion, with co-transporting with facilitated diffusion. So it's how we actually use all the different concentration gradients to help things increase and decrease. So that is it for cell membranes pretty much. What we will do, stop share. What we will do is next week we will have a look at